will be a worksheet. All right, we kind of ended with mutations last week, so I just wanted to go back and review it and do some questions related to mutations. All right, so I gave you this slide here. I did add in one thing. I wanted to add in the terminology of a point mutation. All right, so that's a mutation of a singular nucleotide. All right, so also known as a substitution. So these three here would all be point mutations because it's changing a single nucleotide. All right, and just to review what we're looking at here, we have these silent changes. It's going to cause no change in the amino acid because remember amino acids are conservative, so the first two letters of those codons are generally going to be the same. The third letter is going to differ. So if we have that third letter differing, but it still codes for the same amino acid, right, there's going to be no change in what amino acid it's going to code for after the mutation. We also have missense, right? So that's also a point mutation. We are changing one singular nucleotide base here, but that is changing what amino acid we get as a result. All right, so changing to a different amino acid here. We also have nonsense, right? Again, another point mutation, right? But this time the mutation ends up being a stop. All right, it's telling the cell to stop translating here. All right, and then finally, the other mutation that we had was a frame shift mutation. All right, so this is where we're adding in. We're not substituting anything, but we're adding in another nucleotide, and that's shifting the way that the rest of the sequence is being read. All right, so everything after that addition is going to read as a different nucleotide than what it's supposed to be. All right, so very quickly, what type of mutation is this? Missense. And why is that? Yes, we have a single substitution here, all right? Instead of a T, we have an A, all right? And that now codes for valine instead of, I believe this is glutamine. All right, so one thing I wanted to add with mutations, especially with the missense mutation that you just looked at here, that changes the structure of the protein, so it affects the phenotype and the function, but that's not always a bad thing, all right? So sickle cell anemia is a case of that, all right? If you are a carrier of this anemia, you actually have better defense against malaria, all right? So it's not necessarily always going to be a bad thing. In fact, mutations that end up being a good thing can survive through evolution and eventually become something we see every day. All right, so it's not always a bad thing. All right, I have a question here that's probably going to take you a little bit longer to work through. Work with a partner and figure out what type of mutation am I looking at. So I highlighted this coding strand here. That's what it should look like, but each of these bolded letters represents a mutation.
Where's my page? Mine didn't have a name on it.
Ah, yes, that's right. I totally forgot about that because I had a no-namer.
Mine said like cousins with benefits on the front or something.
That's right. I remember that now.
Because I was in a big rush to get out the door. I was like, oh, I'm going to do that right now.
Want to say? Oh, yeah. Benefits. There we go. That's what it is.
That's what it is.
We can read those bolded letters, right? All right. Once it's up on the screen, it looks much less distinct.
I like that it says cat. The bottom one is French.
Let me highlight these as you're still working. Does anybody need more time, or are we kind of figuring it out? I'm going to assume that means we've figured it out. All right, so let's start with the very first one here. All right, what type of mutation would this lead to? So what's the first, let's see, TAC. If I were to, let's go to the original strand. Let's start there. TAT. What amino acid would that code for in RNA? Yeah, AUA. All right, so AUA. All right, so this ILE molecule here. All right, so if it's ILE, and then all of a sudden we're changing it to what amino acid? AUG. All right, so then all of a sudden it's AUG. All right, so it becomes a start or a methylalanine. All right, so if we're changing amino acids, what type of mutation is it? Missense. Yes, we had it correct. All right, let's start with TGG. What amino acid would that code for? All right, ACC. Oh, where am I? ACC, all right. So if we look at that third column down, it becomes TGT, which would be ACA. Excuse me. The UCA. Oh, I'm confusing myself now. I've been thinking too much about codons. All right, so I would code for AGA would be ACA would be the complementary one, wouldn't it? Because if we're turning this into RNA, this is going to turn into AGA eventually. All right, how about we skip this question and move on to the next one? Because I think this is just confusing you more than it should. And it's starting to confuse myself, all right? All these letters and matching them up with the correct amino acids, all right? I think you get the gist of what I'm trying to say here, all right? So, missense, different amino acid. What would be a silent mutation? We had a silent mutation where we have the same or a different amino acid. Same, all right. What about a nonsense mutation? It'd be shortened because now we're coding for what? To stop, yes. So that would be a nonsense mutation. And then this last one should be a fairly easy one to interpret. All right, you don't even have to look at the mutation per se. All right, but we know it's added in something. So what kind of mutation? Frame shift, yes. So we can go back and work through this, but all the answers should be silent, missense, nonsense, or frame shift. All right? I will not make it this confusing on the exam. I promise. All right. So we just touched on mutation. I'm just going to touch on epigenetics. All right? If I ask you anything on the exam about epigenetics, it's going to be, what is it? All right? I don't need you to know any more specifics. But I do want to look at gene expression. All right, so we have a lot of genes in our DNA, and not all of them are going to be expressed at the same time. All right, so we have the ability for cells to control their level of gene expression to make sure that genes are regulated to ensure proteins are produced at the correct time and at the correct amount. All right, because this is going to save energy in the cell because we're only producing proteins when we need them. All right. Now there is a type of gene called constitutive genes that are always turned on and are always expressed. All right. So when I teach plant biology, I use the example of, if you think of a caterpillar munching on a plant, and that plant has some sort of defense molecule. All right. Maybe it's a molecule that makes the plant taste bitter, and the caterpillar no longer wants to eat it. All right. So some plants only turn on the proteins to make that bitter molecule after it has noticed that the caterpillar has chewed on it, right? Because all the other times, it doesn't need that expressed because there's no caterpillars chewing on it, right? But we have some plants that constitutively make that bitter molecule. They just always have it, right? So the first time that caterpillar munches into the leaf, it's going to get that defense molecule because it's always being produced. All right, so we have those that are always expressed, but we have a lot more that are only expressed when we need them. All right, so this is very important for cell differentiation. All right, so think about how many different cells we have in our bodies. All right, we all have the same genome in every cell, but the way that the genes are expressed are going to determine what that cell becomes. All right, so maybe that cell is a muscle cell, or a pancreas cell, or a blood cell. All right, so you have all the same genome, but express different proteomes. All right, so we're going to express different levels of proteins, or maybe different proteins altogether. All right, so a proteome is what types and how much of a protein is being expressed in each cell. All right, so the example that I'm going to give for eugenics, all right, and how the environment can affect our level of gene expression, all right, is looking at this bacteria, E. coli, and lactose. All right, so when lactose is available in its environment, the bacteria, E. coli, is going to make two proteins, all right, lactose permease or beta-galactosidase. All right, one's going to transport lactose into the cell. One is going to break down the lactose. All right, now when lactose levels drop, these proteins are not going to be made, or they'll start to degenerate. All right, so I have this nice image up here. We can see lactose becoming available as these triangles outside of the cell. All right, as it becomes available, we notice that these two proteins that the cell is making multiplies, and we have many, many more of them. All right, they're breaking down that lactose. Once lactose is gone, all right, these proteins are going to degrade because we don't need them anymore. All right, so that's a good example of how the environment is influencing gene expression. And that's the whole concept of epigenetics. All right, so answer these two questions with each other. And I'm going to pass around a worksheet that you can start on. And then we'll come back and answer these. What's your major? Potentially.
Let's take a look.
All right.
I just need physics next year. That sounds disgusting. I hate physics. What's your hot take on physics? Physics or chemistry?
Actually, never taken physics.
How'd you get with that?
I took a astronomy class that I can counted tolerate. I honestly don't know how I got away with it. It's a disadvantage in life.
Well, I should look into seeing if that loophole is still there.
All right, so how does gene expression lead to cell differentiation? How do we get a muscle cell versus a pancreas cell? Yeah.
Genes express different proteins, then determine what kind of cell it will be.
Yeah, we're expressing different proteomes. They have the same DNA, but they are expressing different genes in different amounts and types of proteins, which leads to cell differentiation. All right. What are some other reasons that gene regulation is important? I didn't specifically talk about it, but we can make some guesses. Cancer? Yeah. I'm not a cancer biologist, but if we mess up our gene regulation, we can totally get some cancerous cells going there. Any other types of reasons that it's important? Stem cell research, yeah, especially. So stem cell research is where we're looking at how cells can differentiate, right? Let's see, another reason it might be important, saving energy, all right? So we're not always using our energy to produce proteins we don't need. Might be important to only turn on the genes that we need in certain situations, right? So think about maybe your roommate jumped around the corner and scared you and you felt a bolt of adrenaline, all right? Think about if that adrenaline was present all the time. All right, that'd be a lot of adrenaline we'd have to constantly make. Probably wouldn't make us feel so great. All right, so making sure we only need molecules when we need them. All right, anything else anyone would like to add? All right, so the rest of the day I kind of have budgeted out as a worksheet day because I feel like we had to go through a lot of material really fast. So this worksheet has a bunch of questions that you can work through specifically related to transcription and translation. And then our review session on Monday, I'll have some other questions that we can talk about during class. Otherwise, bring your own questions and I will answer them. If you have not submitted that resource activity where you post a link to a resource, that is due by today.
Check out my resource.
All right, I will check it out. And then assignment 3, due on Monday by midnight.
Or 12.
All right, so go ahead and work through that worksheet. And if you have any questions, let me know.